Hi everyone, today I'm going to talk about the paper titled Robust and Efficient Approach to Diagnose Sickle Cell Anemia in the Blood. This paper has been accepted in the International Conference on Intelligent Systems Design and Applications. The authors of this paper are Leif Alsapatu gonna be the speaker, and the first author of this paper Dr. Muhammad, Dr. Omran, and Dr. Zhang Lin. And this paper in cooperation between Faculty of Science and Engineering, Queensland University of Technology, Brisbane, Australia, and the University of Information Technology and Communications, Baghdad, Iraq. So what is sickle cell anemia? So sickle cells is that when the red blood cells in the normal shape, they are in circular shape, just like that, this shape. But when these cells infected by this disease of sickle cell anemia will be in the sickle shape. When they are in the sickle shape, they stuck in the blood vessels like that, and they lead to the different problems of like stroke or different problem of blood. So the aim of this paper is to count the number of normal and abnormal red blood cells. It's like the sickle cell anemia number and normal blood cells. By doing that will help the doctors to evaluate the level of anemia danger. And the motivation of this paper is in 2015 there are about 4.4 million people infected with the sickle cell disease, while there is 43 million have sickle cells as transporters. The contributions of this paper are first, we propose a new robust approach to classify red blood cells into groups, normal and abnormal red blood cells, based on the area and eccentricity of each cells. Then we count the total number of normal and abnormal red blood cells individually. For the sake of the comparison, we also implement the latest in sickle cell research, which is circular half transform. Then we compare our approach to the circular half transform. So basically, first thing we need to go through the steps. The first step is this is the pipeline of the proposed approach by doing input image, grayscale, and canny edge detection. Then we calculate the area and centricity for each cells. We remove the non-blood cells, then we classify and counting. Then we get the number of normal blood cells and number of abnormal red blood cells. So we will explain in details step by step. The first thing is input image. Let's see the input image. We go back to the paper. This is the input image. As you see, it's just a small smear of red blood cells. These are the normal blood cells, and these are the sickles. We just took the very small red blood cells smear, and in usual images there are thousands, millions of cells, but we took very small to test, and then we compare it to the bigger samples. Then we check it on the bigger samples. So these are the sickle cells anemia, as you see here. These are, and these the rest. And also the second step is to convert input image to the grayscale image. So to move forward, so we convert it, and then we apply candy edge detection to get the edges, and then we calculate the area and eccentricity for each single cells. So the area is very well known, that's the area of each cells, but the eccentricity for its cells, for some people they have no idea what is eccentricity. So by calculating the eccentricity means if the normal blood cells in the circular shape, the eccentricity is 0 or almost approximate to the 0, like 0.1, 0.2. If the eccentricity is 1, then it's circular in the sickle shape. So it's like measure how much the cells like close to the circular shape. So for example, we have here, if we go down, here is normal blood cells, so the eccentricity of 0.2, while the sickle cells, the eccentricity is 0.9, so which is almost close to 1, which is almost close to 0. So by doing that, we can distinguish between cells. So we can't just say this is normal and this is abnormal. The second step is after we calculate the area, then the next step, area and eccentricity for each cells, we remove normal blood cells or even the inner like radius of each cells. So if we go back to the paper, we see here we have these inner like the cells, because they are just in kind of the curvy images and circular, but they still have the inner value, like inner radius of these images. So how to reduce these or to get rid of these? Even there are some like small cells which are like other like components of the blood. So we get rid of these by getting rid of small areas. So we apply the threshold to get rid of these small normal blood cells or any other objects, to keep the image just with normal blood cells. After we applied threshold to reduce to get rid of small object, we got a new image with this shape. So in this way, there is no any small objects. We guarantee we have only this or not like red blood cells, normal and abnormal. After that, based on the eccentricity, so first thing we removed the normal red blood cells just by applying the area. Then we classify based on the eccentricity. So if it's close to 0, then it's normal. If it is close to 1, there is abnormal. After we classify, we calculate the number of normal blood cells and number of abnormal blood cells. As you see here, this is the eccentricity as I explained before. The eccentricity for normal is 0. The eccentricity for abnormal, which is sickle cells shape, is 1. The next is circular half transform. The pipeline or the procedure of this algorithm is input image, then grayscale image as we applied before, then we remove the noise cancellation by applying different filters, then canny edge detection to get the edge of cells, then we apply circular half transform algorithm to get the number of normal blood cells. The thing here, what they do in this point, I want just to point here. Here, if you see with the experimental result, so the experimental result for our approach, as you see here, the total number of cells 144, the number of normal cells 131, the number of abnormal cells 113. So, and the time of limitation is 0.2 second. So here is the normal cells, and this is the abnormal cells by just applying a threshold to the eccentricity. So this is the result of our proposed approach. So even by using eccentricity, we can distinguish between cells. So if we can say applying, so for in this case, we applied a threshold of 0.7. This 0.7 is like all the cells are like less than 0.7, then they are normal blood cells, and above the 0.7, these cells are abnormal. So we can distinguish further by applying the eccentricity for like less than 0.3 to guarantee they are normal blood cells, then 0.7 until 1 they are for example sickle cells, and between 0.3 and 0.7 we can say other objects of other red blood cell components, other blood components, sorry. For circular half transform, so this circular half transform detects only circular shapes. As you see here, this is the input image that used with our proposed algorithm. Detect only the circular, it doesn't detect the sickle shape. So how they count the number of abnormal? So they bring, so for most of scientists in this area, they bring a normal blood cells, red blood cells, they detect it with the circular half transform, they calculate the normal, then they bring the same like red blood cells smear in same environment and dimensions, and then they calculate also the number, then they subtract the total number of this smear, like using 2 smears, and then they subtract the total number of normal from the total number of normal in the infected smear, and they get the approximate number of abnormal cells, which are the sickle cells anemia. We go back to the PowerPoint. See here the difference. Here we have used with our approach only the infected smear. So as I explained before, we have almost accurate number, while here they detect the total number of normal blood cells from like infected healthy blood cells, and then they calculate the normal cells of infected, like we use the same sample, and then they subtract the total number to get approximate number. As you see here, there is big difference between the numbers. This number almost accurate, while this is very far from this number. So they get just only the approximate number of abnormal cells, and so they use 2 smears instead of 1 smear. But with our approach, we use only 1 smear to calculate with accurate number. Another thing is the implementation time. Here is 0.2, as you see here is 10 seconds. So there is big difference between implementations, and due to that they apply 2 smears instead of 1 smear. So the conclusion of that our approach showed that accurate number of abnormal red blood cells compared to the circular half transform or even other algorithm like watershed algorithm and all other algorithms. And also compared to the circular half transform, it's very accurate and even more efficient. So it shows our approach shows good result and even can apply to the real data, and also it's worth to mention that we tested on 10 different images to make sure it works perfectly. So these are the references, and thank you so much.